The next one is the previous one. So I'll introduce myself. My name is Harald Stover, CEO and CSO of Allarta. And our contribution to the field of cell and gene therapy is here we go, that's us, is to try and enable allogeneic cell transplantation, short-term and in particular long-term cell transplantation for chronic endocrine disorders such as type 1 diabetes. To get some of the who's who out of the way, we are led by our board of directors, Ian Delaney and Mark Campbell, that represent large family offices in Toronto. And our advisory board includes some of the lead polymer scientists in North America, as well as Trevor Reichman, who is a lead islet transplant surgeon at UHN in Toronto. The company was founded by Maria, my co-founder, who has background in strategic partnerships and philanthropic work, and myself about 6 years ago. The technology originally comes out of McMaster University and was taken over into the company where it's been developed very rapidly since. So our basic thesis is that when you talk about cell transplantation into long-term recipients for a number of different indications, we believe that the cells can benefit from being encapsulated in a semi-permeable hydrogel that can deflect immune components of different immune responses, allogeneic, autologous immune responses, with a hydrogel shown in blue around the green cells being permeable enough to allow indiffusion of nutrients and oxygen, in particular glucose in a type 1 diabetes context, and of course open enough for outdiffusion of insulin and any other therapeutic products that may be coming out of these capsules, while deflecting cellular and molecular immune components. So the secret to our technology are our synthetic hydrogels that we can shape into different geometries. They're designed to be immunoprotective, easily processable into different geometries, and retrievable and tunable. We call them life spheres, life lines, life rafts. They all have in common that the cells are never more than about 200 micrometers away from the interface with tissue to allow for good diffusive metabolic connection over that last mile, if you like. This video illustrates how we assemble our synthetic gel formers, which are the key to our technology, with different kinds of cellular payloads. These could be stem cell-derived or donor islets, as well as other cell types. The gel formers are designed to self-cross-link and can then be coated to control permeability, molecular weight cutoff, etc. We shape them into these different geometries, and they all have in common that cells cannot escape from these cross-linked networks, but at the same time, no immune cells can migrate in either. And yet the hydrogel is permeable enough, as I mentioned, for indiffusion of metabolic components and, of course, outdiffusion of the therapeutic payload. At the same time, we design the surfaces inherently and through simple coating, post-processing, to deflect immune components. So overall, this represents a platform for packaging different kinds of cells in an immune suppression-free package that's retrievable and can last for years in vivo. These slides illustrate some of the attributes of these materials. The life spheres, about 1 millimeter in diameter, can be compressed against a point, and they will jump back into their original shape, and that bodes well for long-term implantation in sub-Q or other locations. The lifelines in the center, this is a slowed-down video of human donor islets coming out into a gelling bath with the islets centered in the string. We can extrude multi-meters within about 2 seconds. And on the right, our most exciting technology, which are life rafts, 3D printed strings containing islets or other cells that are implanted sub-Q and can be vascularized rapidly in situ. These are retrievable devices. The key to this technology is that we focus on designing the gel formers to do much of the heavy lifting in terms of building the design, cross-linking, and the immune evasiveness. So the processing, whether it's capsules, lifelines, or life rafts, can be done in a centralized facility or in principle also on-site in a hospital upon delivery of cells. In type 1 diabetes, the preclinical proof of concept involves immunocompetent STZ diabetic rodents and larger animals. In the top panel, you'll see our transplantation of fairly marginal loads, about 1,000 IEQ, of rat donor islets into STZ diabetic black 6 mice. These are fully immunocompetent mice, and you see blood sugar control out to about a year, which is about as long as you want to go in a mouse. On the top right, an allogeneic transplant, again, of rat islets into STZ diabetic V-star rats. Our ultimate payload, of course, will be human beta cells. So on the bottom left, you see human donor islets implanted into the same STZ diabetic black 6 mice. On the red line, you see the blood sugar control that goes out to about 50 days. In contrast, the gray line shows free donor islets, human donor islets, in this xenogenic transplant. They do not affect any blood sugar control. And on the bottom right, a study that was funded by Breakthrough T1D, formerly JDRF, where we expanded this work into Yorkshire pigs, healthy and diabetic. We saw blood sugar control with marginal doses of human donor islets and C-peptide and good islet viability for a couple of weeks. This slide shows an example of live rafts, and to illustrate, we can encapsulate a number of different cell types. We are talking here about human mesenchymal stromal cells in cluster form that are encapsulated. You see viability on day 0, day 1, day 3, pre-implant, and then on the right-hand side, 4 weeks after explant from immunocompetent rats with good viability. In addition to addressing transplantation challenges into patients, we are also aiming to address other logistical challenges in the therapeutic cell field. 2 of them I'll talk to here. One of them is the fact that we all need to cryoprotect cells and ship cells for sometimes multiple days. DMSO has been used for about 60 years in this field, and while it works, it's not ideal for cells, for patients, or ultimately for caregivers. So we believe as cell therapies start to scale, we need to develop additional, improved, less harmful ways for the backroom operations of cryoprotecting cells. At Allarta, we've developed a microgel formulation that tries to replicate the function of DMSO with very little or no DMSO or other organic cryoprotective solvents. The Allarta cryogels are shown in red. They are fluorescently labeled for visibility. They can be packed into a jammed gel, shown in the top left and in the inverted vial. They are fully transparent. We embed different cell types into them. And the microgels, as I will show in a second, they will pack very closely around the cells, dehydrate the cells for internal cryoprotection, and allow storage both at room temperature at 4 degrees and minus 80 degrees, and liquid nitrogen, of course. They're cell compatible. They stay outside of cells, so don't interfere with the internal biochemistry. They're anti-fouling, scalable. We can make them degradable, and they work for multiple different cell types. The mode of action is shown here. The blue microgels conform tightly around embedded cells. They fill all the interstitial volumes, so there's no chance for ice crystal formation that would otherwise damage cell walls during cryostorage. When we thaw them, we can either wash away the microgels or degrade them. They're also biodegradable. And so we believe this is an important contribution for logistics in cell therapies as we go towards scaling. On the left-hand side, one example where we store human donor islets in these fully transparent gels in 3 dimensions. You can think of cars in a parking garage. These are human donor islets in a confocal 3D image. In the middle, iPSC clusters that are cryoprotected and thawed with good viability. On the right-hand side, individual cells, suspension cells, brain endothelial cells, and NK cells that were frozen and thawed with higher post-thaw viability compared with the current standards involving 10% DMSO. This work on the right-hand side was done by an external validating partner, the National Research Council of Canada. This last slide on this subject shows examples of MSCs cryoprotected in our Allarta cryogel. And basically the take-home message from the graph at the bottom left is that the red dots show what happens without any cryoprotectant, including DMSO, so poor viability and poor recovery. And the green and the blue dots crowded in the top right corner show that our formulations with 0 or 2% DMSO are comparable to 10% cryoprotective media. And we do this at lower toxicity to the cells, because DMSO always elicits a toll on cell viability. And on the very right-hand side, we show in blue post-thaw expansion of cells at a rate that's comparable to 10% DMSO. One last slide to show the same thing about iPSC clusters. Basically, small iPSC clusters can be frozen without any problem with post-thaw viabilities comparable to what we see in 10% DMSO. And we're working on scaling this to larger cell clusters, for which there isn't currently an effective cryoprotective solution at scale. The last logistics challenge I want to talk about is scaling of cell therapies. We're aware that our cross-linked capsules could serve as micro bioreactors for expansion and perhaps differentiation of iPSCs. On the top left, we just show basic examples of capsules that were seeded with small amounts of iPSCs, which then expanded to fill the capsules without escape from the capsules. The cross-linked nature of these capsules acts as a little bit of a straitjacket, so prevents both cell escape as well as cell ingress, which is important for safety and therapeutic efficacy. And at the same time, on the right-hand side, we show that these capsules are compatible with other safety features, such as suicide genes that might be triggered by ganciclovir or other suicide triggers. This is work that was done in collaboration with Pancella, which is now part of Pluristics in Seattle. We encapsulated iPSCs fitted with a suicide gene. We watched the cells expand and then ablated them through multiple applications of ganciclovir. So an additional safety factor for these potential therapies. Allarta is a vertically integrated company. We are rooted in polymer science, which is where the initial IP and our current IP is based. We are housed in a 6500 Class A set of biolabs. We have multiple cell biology labs. We have a general polymer biomaterials lab, our own analytical facility. And the polymer chemists are supported, of course, by engineers that do the hydrogel printing, cell biologists to look after the cells, and we have our own sizable preclinical team. So we can learn iteratively very quickly as we go through these processes. I'll sum up by saying I hope I've been able to show you a little bit about how we use a molecular-based approach to build retrievable encapsulation devices for cells and other cell support materials. These are retrievable materials that provide extra safety, are easy to handle. You always know where they are. We are developing plans for scale-up and manufacturing, dealing with logistical issues as we move towards the clinic in perhaps late 26 or early 27. With that, I want to thank the organizers again to put up a really beautiful second time in a row conference here in Rome, and thank you for your attention.
Thank you. Our next presentation is Cellistic.